Chapter 1. Cell Theory. Prior to the 1600s, organisms were perceived as being complete and inseparable into smaller parts. This was due in part to the inability to see smaller structures through simple optical instruments like magnifying glasses. In 1665, Robert Hooke assembled a crude compound microscope and tested its properties on a piece of cork. He noticed a honeycomb-like structure and compared the spaces within the cork to the small rooms of a monastery known as cells. Because cork consisted of desiccated non-living cells, Hooke was not able to see nuclei, organelles, or cell membranes. In 1674, Anton von Leeuwenhoek was the first to view a living cell under a microscope. Later, researchers noted that cells would be separated and that each cell was a distinct structure. Further research indicated that tissues were made of cells, and the function of a tissue was dependent upon the function of the cells that make up the tissue. 2 centuries later, in 1850, Rudolf Virchow demonstrated that diseased cells could arise from normal cells in normal tissues. The original form of the cell theory consisted of 3 basic tenets. All living things are composed of cells. The cell is the basic functional unit of life, and cells arise only from pre-existing cells. Through advances in molecular biology, a 4th tenet has been added to the theory. Cells carry genetic information in the form of deoxyribonucleic acid, or DNA. This genetic material is passed on from parent to daughter cell. Cell theory has created an interesting dilemma with respect to viruses. Viruses are small structures that contain genetic material but are unable to reproduce on their own. This violates the 3rd and 4th tenets of the cell theory because virions can only replicate by invading other organisms and because they may use ribonucleic acid, or RNA, as their genetic information. Therefore, viruses, discussed later in this chapter, are not considered living organisms. Part 2. Eukaryotic cells. The first major distinction we can make between living organisms is whether they are composed of prokaryotic or eukaryotic cells. Prokaryotic organisms are always single-celled, while eukaryotic organisms can be unicellular or multicellular. Whereas eukaryotic cells contain a true nucleus enclosed in a membrane, prokaryotic cells do not contain a nucleus. The major organelles are identified in a eukaryotic cell. Golgi apparatus, endoplasmic reticulum, ribosomes, cytoplasm, mitochondrion, lysosome, nuclear membrane, cell membrane, endocytotic vesicle, centrioles, nucleolus, nucleus. Membrane-bound organelles. Each cell has a membrane enclosing a semifluid cytosol in which the organelles are suspended. In eukaryotic cells, most organelles are membrane-bound, allowing for compartmentalization of functions. Membranes of eukaryotic cells consist of a phospholipid bilayer. This membrane is unique in that its surfaces are hydrophilic, electrostatically interacting with the aqueous environments inside and outside of the cell, while its inner portion is hydrophobic, which helps to provide a highly selective barrier between the interior of the cell and the external environment. The cell membrane is such an important topic on the MCAT that an entire chapter is devoted solely to discussing the structure and physiology of biological membranes. The cytosol allows for diffusion of molecules throughout the cell. Within the nucleus, genetic material is encoded in deoxyribonucleic acid, or DNA, which is organized into chromosomes. Eukaryotic cells reproduce by mitosis, allowing for the formation of two identical daughter cells. The nucleus. As the control center of the cell, the nucleus is the most heavily tested organelle on the MCAT. It contains all of the genetic material necessary for replication of the cell. The nucleus is surrounded by the nuclear membrane, or envelope, a double membrane that maintains a nuclear environment separate and distinct from the cytoplasm. Nuclear pores in the nuclear membrane allow selective two-way exchange of material between the cytoplasm and the nucleus. The genetic material, or DNA, contains coding regions called genes. Linear DNA is wound around organizing proteins known as histones, and is then further wound into linear strains called chromosomes. The location of DNA in the nucleus permits the compartmentalization of DNA transcription separate from RNA translation. Finally, there is a subsection of the nucleus known as the nucleolus, where the ribosomal RNA, or rRNA, is synthesized. The nucleolus actually takes up approximately 25% of the volume of the entire nucleus and can often be identified as a darker spot in the nucleus. Mitochondria. Mitochondria are often called the power plants of the cell in reference to their important metabolic functions. The mitochondrion contains two layers, the outer and inner membranes. The outer membrane serves as a barrier between the cytosol and the inner environment of the mitochondrion. The inner membrane, which is arranged into numerous enfoldings called cristae, contains the molecules and enzymes of the electron transport chain. The cristae are highly convoluted structures that increase the surface area available for electron transport chain enzymes. The space between the inner and outer membranes is called the intermembrane space. The space inside the inner membrane is called the mitochondrial matrix. The pumping of protons from the mitochondrial matrix to the inner membrane space establishes the proton motive force. Ultimately, these protons flow through ATP synthase to generate ATP during oxidative phosphorylation. Mitochondria are different from other parts of the cell in that they are semi-autonomous. They contain some of their own genes and replicate independently of the nucleus via binary fission. As such, they are paradigmatic examples of cytoplasmic or extranuclear inheritance, the transmission of genetic material independent of the nucleus. Mitochondria are thought to have originated when the engulfing of an aerobic prokaryote by an aerobic prokaryote resulted in a symbiotic relationship. Mitochondrial structure. Matrix. Inner membrane. Mitochondrial DNA. Molecular complex. Energy-producing machinery. Abundant ATP powers cellular activities. In addition to keeping the cell alive by providing energy, the mitochondria are also capable of killing the cell by release of enzymes from the electron transport chain. This release kickstarts a process known as apoptosis, or programmed cell death. Lysosomes. Lysosomes are membrane-bound structures containing hydrolytic enzymes that are capable of breaking down many different substrates, including substances ingested by endocytosis, and cellular waste products. Lysosomes often function in conjunction with endosomes, which transport, package, and sort cell material traveling to and from the membrane. Endosomes are capable of transporting materials to the trans-Golgi, to the cell membrane, or to the lysosomal pathway for degradation. The lysosomal membrane sequesters these enzymes to prevent damage to the cell. However, release of these enzymes can occur in a process known as autolysis. Like mitochondria, when lysosomes release their hydrolytic enzymes, it results in apoptosis. In this case, the released enzymes directly lead to the degradation of cellular components. Endoplasmic reticulum. The endoplasmic reticulum, or ER, is a series of interconnected membranes that are actually contiguous with the nuclear envelope. The double membrane of the endoplasmic reticulum is folded into numerous invaginations, creating complex structures with a central lumen. There are two varieties of ER, smooth and rough. The rough ER, or RER, is studded with ribosomes, which permit the translation of proteins destined for secretion into the lumen. On the other hand, the smooth ER, or SER, lacks ribosomes and is utilized primarily for lipid synthesis, such as the phospholipids in the cell membrane, and the detoxification of certain drugs and poisons. The SER also transports proteins from the RER to the Golgi apparatus. The Golgi apparatus. The Golgi apparatus consists of stacked membrane-bound sacs. Materials from the ER are transferred to the Golgi apparatus in vesicles. Once inside the Golgi apparatus, these cellular products may be modified by the addition of groups like carbohydrates, phosphates, and sulfates. The Golgi apparatus may also modify cellular products through the introduction of signal sequences, which direct the delivery of the product to a specific cellular location. After modification and sorting in the Golgi apparatus, cellular products are repackaged in vesicles, which are then directed to the correct cellular location. If the product is destined for secretion, the secretory vesicle merges with the cell membrane and its contents are released via exocytosis. Peroxisomes. Peroxisomes contain hydrogen peroxide. One of the primary functions of peroxisomes is the breakdown of very long-chain fatty acids via beta oxidation. Peroxisomes participate in the synthesis of phospholipids and contain some of the enzymes involved in the pentose phosphate pathway. The cytoskeleton. The cytoskeleton provides structure to the cell and helps it to maintain its shape. In addition, the cytoskeleton provides a conduit for the transport of materials around the cell. There are three components of the cytoskeleton: microfilaments, microtubules, and intermediate filaments. Microfilaments. Microfilaments are made up of solid polymerized rods of actin. The actin filaments are organized into bundles and networks and are resistant to both compression and fracture, providing protection for the cell. Actin filaments can also use ATP to generate force for movement by interacting with myosin, such as in muscle contraction. Microfilaments also play a role in cytokinesis, or the division of materials between daughter cells. During mitosis, the cleavage furrow is formed from microfilaments, which organize as a ring at the site of division between the two new daughter cells. As the actin filaments within this ring contract, the ring becomes smaller, eventually pinching off the connection between the two daughter cells. Microtubules. Unlike microfilaments, microtubules are hollow polymers of tubulin proteins. Microtubules radiate throughout the cell, providing the primary pathways along which motor proteins like kinesin and dynein carry vesicles. Cilia and flagella are motile structures composed of microtubules. Cilia are projections from a cell that are primarily involved in the movement of materials along the surface of a cell. For example, cilia line the respiratory tract and are involved in the movement of mucus. Flagella are structures involved in the movement of the cell itself, such as the movement of sperm cells through the reproductive tract. Cilia and flagella share the same structure, composed of 9 pairs of microtubules forming an outer ring, with 2 microtubules in the center. This is known as a 9+2 structure and is seen only in eukaryotic organelles of motility. Bacterial flagella have a different structure with a different chemical composition, as discussed later in this chapter. Centrioles are found in a region of the cell called the centrosome. They are the organizing centers for microtubules and are structured as 9 triplets of microtubules with a hollow center. During mitosis, the centrioles migrate to opposite poles of the dividing cell and organize the mitotic spindle. The microtubules emanating from the centrioles attach to the chromosomes via complexes called kinetochores and exert force on the sister chromatids, pulling them apart. Intermediate filaments. Intermediate filaments are a diverse group of filamentous proteins, which includes keratin, desmin, vimentin, and lamins. Many intermediate filaments are involved in cell-cell adhesion or maintenance of the overall integrity of the cytoskeleton. Intermediate filaments are able to withstand a tremendous amount of tension, increasing the structural rigidity of the cell. In addition, intermediate filaments help anchor other organelles, including the nucleus. The identity of the intermediate filament proteins within a cell is specific to the cell and tissue type. Tissue formation. One of the unique characteristics of eukaryotic cells is the formation of tissues with division of labor, as different cells in a tissue may carry out different functions. For example, in the heart, some cells participate in the conduction pathways, while others cause contraction. Still others serve a supportive role, like maintaining structural integrity of the organ. There are 4 tissue types: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. While muscle and nervous tissue are considered more extensively, we explore epithelial and connective tissues below. Epithelial tissue. Epithelial tissues cover the body and line its cavities, providing a means for protection against pathogen invasion and desiccation. In certain organs, epithelial cells are involved in absorption, secretion, and sensation. To remain as one cohesive unit, epithelial cells are tightly joined to each other and to an underlying layer of connective tissue known as the basement membrane. Epithelial cells are highly diverse and serve numerous functions depending on the identity of the organ in which they are found. In most organs, epithelial cells constitute the parenchyma, or the functional parts of the organ. For example, nephrons in the kidney are composed of epithelial cells, and hepatocytes in the liver and acid-producing cells of the stomach are epithelial cells. Epithelial cells are often polarized, meaning that one side faces a lumen, the hollow inside of an organ or tube, or the outside world, while the other side interacts with underlying blood vessels and structural cells. For example, in the small intestine, one side of the cell will be involved in absorption of nutrients from the lumen, while the other side will be involved in releasing those nutrients into circulation for use in the rest of the body. We can classify different epithelia according to the number of layers they have and the shape of their cells. Simple epithelia have one layer of cells. Stratified epithelia have multiple layers. Pseudostratified epithelia appear to have multiple layers due to differences in cell height, but are, in reality, only one layer. Turning to shape, cells may be classified as cuboidal, columnar, or squamous. As their names imply, cuboidal cells are cube-shaped, and columnar cells are long and thin. Squamous cells are flat and scale-like. Connective tissue. Connective tissue supports the body and provides a framework for the epithelial cells to carry out their function. Whereas epithelial cells contribute to the parenchyma of an organ, connective tissues are the main contributors to the stroma, or support structure. Bone, cartilage, tendons, ligaments, adipose tissue, and blood are all examples of connective tissues. Most cells in connective tissues produce and secrete materials such as collagen and elastin to form the extracellular matrix.